Hello, everybody. My name is Tanveer Ahmad. I am postdoc fellow at Kolmogru Lab at NCI at NIH. Today, my talk is about Vahan, which we developed a haplotype-specific copy number profiling tool for cancer genomes using long-read sequencing data. I will just start from structure variations. Many of you know, like we focus on variations in genome related to structure variations or some small variations. Structure variations can affect multiple genes or whole chromosomes. This could be from simple deletion in insertions, duplications to complex rearrangements in form of chromothripsis or chromoplexy. These variations could lead to change in copy number profiles in different segments of the genome. Generally, we can say a couple of kilobase pairs of whole arm of a chromosome in a gain or loss could be called copy number. Structure variations or copy numbers analysis help understanding tumor initiation and progression, could be indicator of treatment resistance and have prognostic and predictive value in cancer. Previously, for short reads, there are a couple of methods have been developed for whole genome sequencing data sets, which are Battenberg, ASCAT, and Hatchet, and Purple, and CNVKit, and there are a list of methods which have been used and developed for short reads. For long reads, Savannah is a one for long reads specifically for whole genome sequencing data, and Purple could be used for long read as well. Reconstructing deployed genome possible through long phase block because of long read sequencing data. And it's also possible now to have chromosome scale phasing in conjunction with long read and high C data. But particularly for Savannah, they have developed this long read based method, but they're not exploiting the benefits of long read, long phase blocks. Vahan is the first haplotype specific copy number tools for long reads. Vahan generates chromosome scale phasing, which is comparable to high C and 4C dataset. So here, this is one example. Both colors represent haplotype 1 and haplotype 2. Although, because of SNV's phasing, we can get very long phase segments, but still there are some switches between those segments, which happens whenever there are reads and there is no informative SNVs, and it breaks the phase blocks. So long read sequencing and after the phasing, it gives somehow like this type of, this is bin coverage of one of the chromosome in one of the cell line. So first we try to make correction in this phasing. The assumption is phase block boundaries and CNA boundaries unlikely to coincide, and minimizing the number of CNS changes or switches within a haplotype. So this is the result of our first phasing extension procedure, which Vahan produced. And through this, you can see very clearly both haplotypes have very clear coverage. And from here, we can easily say like which is in which haplotype one region is LOH, and in other haplotype HP2 some segments, some regions are amplified. I will just go through a little bit about Vahan internal structure. Vahan support both tumor only and tumor normal modes. It as an input, it takes normal phased VCF or tumor phased VCF, as well as their corresponding tumor BAM. We also integrate structure variations call. In the first step, we generate pileup from informative SNVs from the phase VCF file, and we generate this type of bin coverage. This bin coverage is customized, and here we are just showing 50 KB bin size. And at the end, in the step 3, you can see like we can get phase blocks, and there are switch errors in those phase blocks. So as I mentioned previously, we correct these phase blocks, and in the third step, you can see we get more accurate haplotypes coverages. We incorporate SV and breakpoint segment boundaries, which are being impacted by copy number changes. We use autocorrelation to estimate the first copy of the genome, and based on that, we determine all other copies of Gaussian distributions. Subsequently, we assign copy number states. And this is the output of the Vahan. It generates bed files, VCF files, as well as some interactive plots with ploidy and purity estimation, confidence score of different. It produced different solutions, and based on those, we can give confidence score, breakpoints, information, LOH regions, and bin coverage, subclonal and integer copy numbers, and BLE frequency and gene annotation, and so on. So I will just go through this interactive plots step by step. So these are the ploidy and purity estimation of one of the cell line HG008, which is being developed from NIST. This is a breakpoint information. This is the coverage, bin coverage, custom bin size. And these are the copy number, integer copy number states. Here we also show the subclonal copy numbers and BLE frequency and gene annotations. For the results, we have a sequenced Castle with different cell lines. From those panel, I just selected one cell line at H1437. Here we have different coverage levels. We simulated different coverage levels from 60x to 20x coverage level and different purity levels to test the correctness of our method. And we have compared this tool with PacBio tools, Purple and Savannah, and Illumina-based tool, Purple and Hedge and Battenberg. So this is inside Vahan with different coverage levels and with 100% purity. We see like there is no much change, not much that much changes in the copy number of profiles. These are the both haplotype 1 and haplotype 2, and tumor only and tumor normal modes. We have generated truth set considering 100x, 100% purity and 60x as a truth set. This is the comparison with different long read and short read based methods. This is tumor only mode. We have developed a consensus based on all these solutions because there is no available benchmark or truth set for copy numbers for any of the cancer data sets. For this, we have developed consensus, and for this, we are calculating concordance score for each data set, each tool. This is HP1 and this is HP2. We see like Vahan is producing almost equal to Purple or Illumina short read based methods. This is tumor only mode. Many of the tools are struggling with tumor only mode, and we use the same data set as a tumor only. And here Vahan also produce very satisfactory results. Concordance going up to 96 and 98%. Some tools, whenever there is low coverage in cancer data, like 20x, 30x, their performance degrades. So Vahan produces, even with different different coverage levels, it's very consistent in producing copy number calls. Here we show one case in 100% purity and 60x, how different colors are producing different copy numbers increase and decrease. And this is 20x, even it's worse than the previous in 60x coverage. Here I just want to show one example of chromothripsis. So this is another cell line, HCC 1954, where we see like an event of ecDNA. And in these two chromosomes, there is so much chromothripsis or some complex events. If you see in this figure, this is the existing output of the ONT data, and both haplotypes are mixed. And basically, in actual, only one haplotype is being impacted. And this is the results of our phase correction, where we can see easily only one haplotype is impacted as compared to the other haplotype has so much fluctuations. This is the high-C results, and we both, you can compare both. We are producing almost similar phasing as compared to high-C and poor-C data sets. In these type of scenarios, all allele-specific tools, they will struggle because they will assign higher coverage to a major allele and the lower coverage to the minor allele. And these coverages, this profile will be so much fluctuated in all these allele-specific methods. Vacan is already integrated in PacBio workflow for somatic datasets, and we are working with ONT, and it will be available in ONT workflow within few days. Manuscript is in preparation, and it will be available in next few weeks. We in the Kolmogorov lab, we have already developed a method which is a structure variation graph-based method. It's called Severus. We have collaborated with Google for small variation color methods like deep somatic. Vacan is the first haplotype-specific copy number tool for long reads. It generates chromosome scale phasing comparable to high-C and poor-C data. It detects clonal and subclonal copy numbers. It supports both tumor and normal pair modes. It estimates correctly purity and polarity of the sample. We have performed very robust testing on different technologies and different data sets and on different tools, both short read and long read. It will be described in the paper. And thank you so much. It's available on the GitHub.
We have time for some questions. I will get started then with one. I was wondering, in your opinion, according to your intuition, do you think that most of the improvements that you get with your tool came from using long reads or from any other feature in your tool?
I think mainly it's because of phase correction. It helps in if we have contiguous segments as compared to the short read or allele specific. It helps in somehow in detecting the copy number segment correctly.
But do you think that does that mean that if you integrate that phase correction as I don't know, like a pre-processing into other tools, then can you further improve it?
Yeah, for long reads, of course, it will be very helpful. Yeah.
And then did you try instead the opposite, to apply your method to short reads to see how it works?
Yeah. So basically, our method is not developed for short read, but then we have tested, and it's working. But for that, for short read, we need some phased VCF, and short read based VCFs are not that much. We have to use PON data sets. So in that scenario, it still works, but then it's becomes similar to short reads. Yeah.
Wonderful. Any other questions?
Hi, thank you for the talk. Do you know if Vacan can detect MSAI? So where on one haplotype a chromosomal segment is lost, and on the other haplotype it's gained at the same time?
I go. Okay. Yeah.
Here, but on the bottom right is what Vacan that's the output, right?
Yeah, this is the initial Bahan phase correction output.
Yes, but so have you seen examples of the top left where there's actual so there's a switch between these haplotype segments?
Yeah, yeah.
And can Wakan detect it?
So yeah, what we are doing like that. This is the coverage,
Mhm.
and these are the basically whenever there is switch, it means the phase block is being broken.
Okay.
So we get those phase block information there from the phased VCF file, the phase blocks, and we switch them based on our assumption of like the phase block boundaries coincide with the CNA boundary unlikely,
Okay.
and then we just change those boundaries to get some sort of this correction.
Okay. Thank you.
Yeah.
Any other questions? I guess maybe just building up on that question, I think if I'm correct, what she meant is whether like if you have like two samples and you apply your method and you have like a gain in one sample and a gain in the other sample, but affecting two different haplotypes, you can detect that. That is correct?
Yeah. Yeah.
Can you analyze multiple samples
Yeah,
together
yeah,
to detect
yeah.
that? Any other questions? Wonderful. In that case, we can thank the
Thank you.
speaker again.